Hello everyone, welcome back to biology. Today we're going to be talking about genetics part 2 and we're going to get into some pedigrees. So there's a bunch of different types of traits and ways that genes can be passed down to offspring. And because we have autosomes, those non-sex chromosomes, and then we have the sex chromosomes, these traits can either be on the autosomes, which is what we call autosomal, or they can be linked to one of the sex chromosomes, the X or the Y. If they are, we call them sex-linked. With these types of traits, if it's X-linked, then females can be carriers because they can have that trait on both of their X chromosomes. So they could be X capital H, X lowercase h. They could be X lowercase, or X capital, X capital. Whereas with males, since they only have one X chromosome, they're either going to have a lowercase h or they could have an uppercase h. And that's the only options they have. So they're either going to have the trait expressed or they're not. Nothing in between. Whereas with females, they can have the trait expressed or it can not be expressed, but they could be a carrier. So one of the questions here, can a male be a carrier with X-linked traits? No, they cannot be because they only have one X chromosome. And the X chromosome that a male receives has to come from his mother because the Y chromosome came from dad and the X comes from mom. So every X-linked trait that a male receives came from his mother. So we're going to work on a few of these. So it says cross a carrier female with a normal male. So a carrier female, we're going to have X capital, X lowercase, and then a normal male. And this is recessive. So he's going to, since he's normal, he does not have it. So he is going to be X capital H Y. And she will be X capital H, X lowercase h. It says, what are the possibilities of their child being colorblind? Then we're going to create our Punnett square. So we've got a 2 by 2 square for this one. We'll put the female towards the top, so X capital H, X lowercase h on top, X capital and Y going down the side. So then we'll fill it in and see what we get. So now that we filled this in, the first top left square is going to be X capital H, X capital H. The second one on the top right is going to be X capital H lowercase h. Bottom left, X capital H Y. Bottom right, X lowercase h Y. So we got to think about what is the possibilities of their child being colorblind, just in general. Well, to be colorblind, you have to have the recessive trait expressed. And males only have one of these traits because they only have one X chromosome. So we'll look at the females first. Both females have a capital H, at least one. So neither of them will be colorblind. If we look at the males, one has a capital H, one has a lowercase h. So half of the males will be colorblind. The possibility of their child just in general being colorblind, that's going to be 25% because it's 1 in 4. The possibilities of their son being colorblind, well, if they have a son, then he has a 50% chance of being colorblind. What are the possibilities of their daughter being colorblind? Well, that's going to be 0%. And there's the answer there. So, with females, because they have 2 X chromosomes, and they actually can have more than that if there's a genetic abnormality, during their development, the chromosome that is not used will be condensed into what they call a bar body. As of right now, it is thought that the process by which these extra X chromosomes are shut down is random, but perhaps one day there'll be some kind of discovery that shows that maybe it's not so random. But at least that's the thought as of right now. So here's a bunch of different examples where you might have an extra X and what happens to it. Here, even in a male with Klinefelter syndrome, they have an extra X chromosome. So that extra X gets condensed into a bar body. And then just with a regular female with 2 X chromosomes, the extra X chromosome will be condensed into a bar body. So these chromosomes, when we think about inheritance, this is a really interesting example. So with these cats that can have this calico or tortoiseshell coat, with males and females, you'll see a different expression because this type of color and expression on the fur coat is on the X chromosome. So if you have a female, well then a female could be XOXO, and the O's code for orange, or they could be XB, XB, which would make them black, or they can be a mixture of the two. So you could have a female that's XO, XB, and that's how you get this calico or tortoiseshell type appearance. But with males, they're either going to be XBY or XOY, so they're either going to be black or orange, and that's what this slide is showing you here. Some other types of expression, you can have a pleiotropic gene, which has multiple phenotypic expressions because one protein is a part of many biochemical pathways or affects many different activities in the body. So the example here is the Marfan syndrome. So essentially what happens is this particular protein, when it's produced, it's all throughout the body, so it can affect many different areas of the body. So some common symptoms of this disorder, they'll have scoliosis, this sort of curved spine. They might have a protruding or a sunken or concave chest. It can affect their eyes. They generally will be taller than your average person, and then they have some interesting abnormalities with their hands and their wrist. The environment can also affect how these genes are expressed. A good example of this is the Himalayan rabbits. The colder portions of their body, a certain protein is expressed differently than in the warmer portions of their body. So in the colder portions, this expression is different, and it actually produces a black coat color gene is expressed in colder portions of the body. So what you see with these Himalayan rabbits is the extremities will become darker than the other parts of their body. Arctic fox, we see something similar where when it's colder, their fur will turn out to be expressed and it will be white. And in warmer areas, it'll have more pigmentation to it, which in both cases creates a nice little camouflage depending on the environment that they're in. And then Siamese cats, similar to the Himalayan rabbits, colder portions, the gene is expressed differently and it gets darker. That's why their extremities tend to be dark. And one of the benefits to this, think about this rabbit, is the extremities are in a colder, they're experiencing colder temperatures. So being a darker shade on the fur will absorb more energy from the sun, more light, and it's going to get warmer. So it'll actually warm up the extremities in colder environments by having that darker pigmentation in the cooler portions of its body. So it helps regulate its temperature. Then we have other traits that are called polygenic traits. This is when several genes affect the trait. Skin tone is a great example of this, also height. So we have 3 different sets of genes affecting skin tone. So what you end up with is, like if you have 2 sets of genes and you crossed them with each other and they're both heterozygous, well, you would have 2 times 2, 4 different combinations there for gametes. 2 times 2, 4 different possibilities for gametes there. So the size of the Punnett square you would need to make to cross 2 traits would be a 4x4. Well, if you have 3 traits, like we have with skin tone, and the parents were heterozygous for each, well, then you're going to have to do 2 times 2 times 2 would be 8, and then the same for this. So you'd have 8 times 8. You have 64 different options. So you have an 8 by 8 Punnett square. So you have this huge Punnett square. So there are 64 different skin tones. And here, this is only showing like 42 or so. But you can see this gradient of darker to light shades that you can have depending on how the traits are expressed and what the parents have. You don't need a Punnett square this large if the parents are not heterozygous for every trait, which is actually pretty rare. So usually they're going to be homozygous for 1 or 2 of these, maybe all 3, in which case the possibility space that you have there would be reduced. This is just the classic example that I always use, where these twins were born to a father who was from the United Kingdom and a mother from Jamaica, and they must have been maybe heterozygous on a lot of their skin tone traits, because they had some children that were much darker and some that were much lighter skin tones. And these were actually twins that were born with different skin tones. Now, they were fraternal, so it's 2 separate eggs, 2 separate sperm cells. But still, it was interesting to see 2 twins with such different skin tones And when we look at the options for skin tones, well, we have 64. So while the chances of this occurring, having a very dark skin tone and a very light skin tone, are extremely rare, it would be technically possible for two kids in the same family, if their parents were heterozygous for each skin tone trait, to have one child that had a very deep pigmentation and one that was very light. So another expression example that we'll see is epistasis. And for this, we call this the masking effect. This is when you have some type of masking trait that is expressed along with the regular trait. So for example, you could have like regular, capital B is for black, lowercase b is for brown for their fur coat color. But then you also have this other trait that's expressed with it that is, maybe we call it allele E. And if E is present, if it's capital, well then melanin is produced. If it's lowercase e, then no melanin is produced. So then we have to compare these in conjunction with each other to see what kind of offspring it's going to make. So for example, here we have this dog with black coat color, this retriever. Well, for that to happen, it needs at least a capital B. And for melanin to be produced to make it dark, it needs a capital E. Well, to have a chocolate or brown retriever, you need the lowercase b's, homozygous recessive, to be brown, but then it also needs a capital E to actually have pigmentation. So you're going to have capital E in both of these cases. But for the golden retriever, you need the absence of pigmentation. So it doesn't matter if it has a capital B or lowercase b's. If it has lowercase e's, then it's not going to be able to produce pigmentation anyway, and you end up with this golden retriever. So it says cross a heterozygous black-furred, heterozygous melanin lab with a heterozygous black-furred, heterozygous melanin lab. So I'm not going to go through all this, but just to kind of skip ahead, we've got a heterozygous for both sets of traits. So these are our 4 gamete possibilities. And again, the way you figure that out is you look at the parent genotype and you see how many options there are. Capital B, lowercase b, those are 2 different options. So we multiply that by how many options are for trait E, 2 different options. So 2 times 2 gives me 4 different gamete possibilities. Whoops. So I know I have 4 different options. And these are the 4 different ways to combine capital B, lowercase b, capital E, lowercase e, if we're going to make a sperm or an egg cell. So that brings us to here. So we're going to have to make, because it's a 4 by 4, we need a 4 by 4 Punnett square. So then we'll start to fill it out. We'll put one parent's genes up here. And one parent's genes going down the side. Sorry, same genes. They're gamete possibilities, so sperm or egg cells. And then we fill in the inside. And we would do this all the way across until we got to the end. So I'm going to skip ahead where this has already been filled out for us. And this is what it's going to look like. So for this, if we start counting through to try to see what it's going to make, we can look at those genotypes and see what the phenotypes will be. And then we just count to get those percentages. So it has to have at least a capital B to be black, but it also has to have a capital E to be black. So I would go through, and this has already been done for us, but I would look for every cell or box that has at least one capital B and at least one capital E. And if we look at that, that's going to be all of these black squares. They all have at least one capital B, one capital E. Well, then to have a golden retriever, it needs to have lowercase e's, and it doesn't matter what else there is. So to find those, we're going to have pairs of lowercase e's. That's the only way to have no melanin production, so that it ends up being a golden retriever. Well, to be chocolate, they need to have melanin production, so they need at least one capital E, but then they also need to have 2 lowercase b's to have that brown color. So that's going to be these 3. So some other things we'll get into is some pedigrees and how to interpret them and how to tell what we're looking at. So if we're looking at autosomal recessive diseases, autosomal means non-sex linked. Recessive means you have to have the lowercase letters to inherit the disorder. So these are caused by recessive alleles, and that's going to be on the autosomes 1 through 22, not on the sex chromosomes. So in order to express this, an individual must receive 2 of the recessive alleles, and they could be carriers. So we think about eye color. Let's say this is what's going on the left here. Well, maybe he's heterozygous for brown eyes, and she has blue eyes. Well, then the possibilities for their offspring, if you did that Punnett square, you're going to have about half who are going to have brown eyes but be carriers, which we see here, and then half are going to have blue eyes, what we see here. So autosomal recessive diseases, here's a list of a few of those if you'd like to pause and look at what they are and what chromosome they're on. And then we can also have autosomal dominant, and in that case, they just need one of the alleles and they have the disorder. So there are no carriers because if they have one of the alleles, they have it. Autosomal dominant, it's going to be on chromosomes 1 through 22, the non-sex chromosomes. And here are some examples of those disorders. So then we could have sex-linked recessive diseases. So if it's sex-linked, it's going to be linked to either the X or the Y. Generally, we're going to use this to refer to those that are X-linked, but technically you would want to say X-linked recessive disease. So females can be carriers, but males cannot because it's linked to an X chromosome. So a female can have one of each, but a male, because he's XY, is either going to have X capital H Y, X lowercase h y, or whatever trait letter we're using, so they can't be carriers. So we're going to apply all that Punnett square that we've been going through with Genetics 1 and Genetics Part 2 to apply that to pedigrees and to interpret pedigrees. The way to interpret a pedigree is it's just a family tree. Squares represent males, so we have the great-grandfather right here. Circles represent females. If it's shaded in, that means they have the disorder or the trait or whatever it is that we're talking about. And if it's not shaded, that means they do not have or they're not affected by the trait. Sometimes they'll half shade a circle to show you that they're a carrier. Sometimes they will not half shade circles to show you that they're carriers. It just depends on the question that they're asking. So you have to interpret them all independently. So recessive pedigrees often skip generations. That's one way to tell if you have a recessive pedigree. To tell if it's autosomal or if it's sex-linked is you can look and see are males and females affected differently. In this case, you have 2 females and 2 males. So that's even effect on both, so it's probably not sex-linked. And then autosomal, because it's not affecting males and females differently. And recessive, it has to be recessive because it skips a generation. Sometimes recessive traits just happen not to skip generations. So you just have to take them each one at a time to see what's going on. If we have euploidy or aneuploidy, euploidy means the normal number of chromosomes. Aneuploidy means there's an abnormal number. So the other thing we'll also look at here is karyotypes, which is a picture of someone's chromosomes. And when you look through at every number, they should have a pair of chromosomes. There should be 2 because there's 23 pairs. So you can look through to see if there's any genetic abnormalities in terms of chromosome number. If there's only 1 chromosome in a place where there should be 2, they call that monosomy. If there's 3 instead of 2, they call that trisomy. So for example, here they have 3, so that would be trisomy 21 because it's in the 21st position. You'll notice they're ranked by length, with the exception of the sex chromosomes. They're going from largest to smallest. So someone with Downs would be chromosome 21. They have 3 of those instead of 2. Now, why does that happen? Well, it's generally caused, in this case, in terms of chromosome number, from non-disjunction. So during meiosis, when the chromosomes line up at the metaphase plate and they're connected to the spindles and they're trying to be pulled apart, if they do not separate, well, then you end up with too much DNA in a couple of sperm or egg cells and too little DNA in a couple of those gametes. So that's going to affect the chromosome number when that sperm or egg cell is fused with its complementary gamete. And here are some disorders caused by non-disjunction. Some other things that can happen is genes can be deleted, they can be duplicated, their order can be reversed, or they can even be put in the wrong place. And this can happen during crossing over, prophase 1 of meiosis 1. And here are some examples of what that might look like. A whole gene could get deleted, or maybe a gene gets duplicated, and then you end up with too much, or the genes themselves could be swapped, the order, they could be inverted, and then they might move into the wrong location. So I'm going to have that translocation. Okay, so now we're going to get into, I'll show you a couple karyotype things first, like what even is that? It's just a picture of a person's chromosomes, and they have a couple of different processes to try to sample a baby in its mother's womb, to try to sample its genetics, its chromosome number, to see if it's going to be born with a chromosome number abnormality. And what they can do is they have a couple of different ways of getting that amniotic fluid, because this organism is going to be shedding skin cells. So if you can get some of that amniotic fluid, then you can test the cells to figure out how many chromosomes they have and if there's any abnormalities there. So there's an example of them pulling out the fluid. There's a couple of different ways they can get in there to get it. There is a risk with these kind of procedures, because if you puncture that amniotic sac and the fluid, you lose a lot of fluid, well, then the baby's not going to have the right amount of fluid for its muscles to develop. All right, so let's go through some pedigrees. So again, the basic idea is circles are females, squares are males, lines between them represent a union, and then lines going down represent the offspring that union has produced. So in this case, everything connected to them directly is going to be their offspring. So these two parents had a daughter, a son, and another daughter. So they had 3 kids, 2 daughters, and 1 son. Also, the generation number you'll see listed on the left-hand side here. They usually use Roman numerals 1, 2, 3, 4. Just the question for an example. How many children did generation 2, 1, and 2, generation 2, individuals 1 and 2, they would have 1 child, and it would be a girl. Okay, here's another pedigree. So if it's autosomal recessive, so that means non-sex chromosomes, so it's not transmitted through the sex chromosomes. It's an autosomal trait, and it's recessive. So we're thinking lowercase letters. One of the easiest ways to determine if it is an autosomal recessive pedigree is to see if there is an individual that has the disease, but neither of their parents have the disease. So if we look here, these 2 parents, their 3 kids, none of their kids have the disease, but their dad does. So we know that's a recessive trait. If we look to see, well, is it sex-linked? The males, there are 4 males who have it, and there are 5 females who have it. Basically the same, so it's not going to be sex-linked either. If it's autosomal dominant, the easy way to determine if it is this is every individual that has the disease must have a parent with the disease, not just once, every single time. So if we look here at this person, one of their parents has the disease. This person as well. Everyone that has the disease has a parent, at least one parent, who has the disease. So that's one way to tell if it's autosomal dominant, because again, they just have to get 1 letter and then they have it. So one of their parents would have to have had it expressed for them to even get that capital letter or that allele. So if it's sex-linked or X-linked, one of the easy ways to determine it, if it is sex-linked, is if males and females are affected differently. So if we look here, we'll notice almost all the females are carriers. We only have 1 female who actually has the disorder, and all the males have it. So there are no male carriers. Definitely going to be sex-linked, and it has to be X-linked, because males are only getting 1 X chromosome, so they either have it or they don't. So we'll go through some examples. It says, how many children do individuals 1 and 2 have? How many generations are shown in this pedigree? So this is a basic question before we get to diagnosing. How many children do individuals 1 and 2 have? They would have 3. How many generations are shown? 1, 2, 3 generations. What is the form of inheritance shown in this pedigree? So pause the video, see if you can figure it out, and then we'll check your answer. So it should have been autosomal recessive, so non-sex linked and a recessive trait. And again, females and males affected at the same rate, and it skipped a generation. So autosomal recessive. Try to figure this one out. It says, what form of inheritance is shown in this pedigree? Pause the video and then check your answer. So it should have been autosomal dominant. In this case, every person who has the disease has a parent, at least one parent, who also has the disease. So that's going to be a dominant trait. Autosomal because males and females are affected evenly, 3-3. If it's off by 1, it could still be non-sex linked, could be autosomal. Really, there's going to be a big difference usually when it's sex linked, especially on a big pedigree. And then karyotypes, just go through some examples here so you guys know how to recognize what's going on. This right here would be a normal karyotype because every chromosome has a partner. With the exception of the sex chromosomes, you should only have 2 sex chromosomes for it to be, pretty like a normal number of chromosomes, which would be 46. So anything outside of that we say is abnormal. So in this case, this is still normal because it's 1X, 1Y. So you need to be able to tell, is it a male or female? And then are there any chromosome abnormalities? So in this case would be male. There are a pair of chromosomes at every location. So there are no genetic abnormalities, at least in terms of chromosome number. So if we look at this one, well, they have an extra 21st chromosome. So they have trisomy 21, which is also known as Down syndrome. In this one, they have 2X and 1Y. They actually call that Klinefelter syndrome. And in this case, the symptoms are you have an X chromosome, so they're going to have more estrogen. And these will develop as males, but they'll have potentially breast development and a lot of side effects, extra fat on their body, and they usually take some type of hormone replacement therapy to alleviate some of the symptoms of Klinefelters. And a lot of times they'll be infertile. Then 2Y, so still going to be a male. They call this super male syndrome. Everything else is the normal number except for an extra Y chromosome. In this case is Turner syndrome, just 1X instead of 2, and there's no Y chromosome here. So that would be the abnormality there, is that there's only 1X instead of 2. This one, an extra X, so they call that a meta female. So again, just being able to recognize abnormalities in chromosome number and being able to interpret those pedigrees. Thank you.